Today, I'd like to tell you the story that we've been working on for the last 10 years, the taxonomy of the Penstemon Scariosis complex. We are in the later steps of a peer review process for future publication in the Western North American Naturalist. The Penstemon Scariosis complex is found in central Utah up through the northeastern quadrant and in the adjacent areas of Colorado and Wyoming. There are 4 varieties involved. In order to get a better understanding of the historic view of Penstemon Scariosis, we need to understand where they have been viewed each variety to be from. Alba fluvis from the Uinta Basin region, Cyanomontanus from the Blue Mountain area. Goridii has overlap with each of the other varieties and has greatest geographic range. Finally, variety Scariosus has been found in the southern areas of where Penstemon Scariosis range is at. To understand this complex further and to try to understand the relationships of each of those varieties from a genetic point of view, we initiated a population genetic structure study. We used 10 molecular markers, often referred to as a genetic fingerprint. 66 accessions were collected across all 4 recognized varieties across the entire geographic range. We included 4 accessions of Penstemon subglaber as genetic outliers in the initial analysis. To understand how to read the results of our first study, one must understand that each color in the bar graph represents a related population. A bar with mixed colors represents the percent of time a sample segregates from a specific population during structure statistical analysis. We learned 3 things in this study. First, Penstemon subglaber is distinct, as expected. Penstemon Scariosis variety Alba fluvis is genetically distinct. There's no geographic overlap with other varieties. We recommend returning variety Alba fluvis to a species status. Third, the Tabby Mountain population was a complete surprise. We initially found a small group of plants, or rather Jolynn Stevens found it in 2014 and brought it to our attention. In 2016, because of studies that took place in those intervening years, we went back and searched a large geographic range and collected many more samples. We next focused our studies by removing Penstemon subglaber and Penstemon alba fluvis from further analysis. Here we present the first of 2 focus studies. In this study, we looked at the structure of the remaining samples. First, we must understand that the breaks of Cyanomontanus, Goridii, and Scariosis, and Tabby Mountain are based strictly upon morphometric characteristics, with the exception of Tabby Mountain, and not geographic. We learned 3 things by studying this way. First, there is a lot of genetic admixture and incorrect variety assignment in the historic populations of variety Goridii and Scariosis. We can especially see that when we finally map it out and begin to look at each of these bars as a pie chart in geography, where the Scariosis, the Goridii, and the cyanomontanous type locations are at. Note that the cyanomontanous area appears to be much larger than the small circle we originally presented as being the original zone for cyanomontanous. If we expand that out, it's a lot larger region. Finally, the third thing that we get out of this is a genetic distinct population associated with the Tabby Mountain area, which is expanded out in the lower right. In our second focus study, we use principal coordinate analysis, or PCOA. The colors in the PCOA represent a strict historical morphometric classification assignment. SCA 062 and 015 are less than 2 miles apart, and yet they are genetically unique from each other. SCA 013 segregates with cyanomontanous in structure analysis, and morphometrically they have glandular hairs. It is an area that we refer to as an admixture location. Let's take a look at the PCOA in a third dimension, the two-dimensional plot here, a three-dimensional plot, and the second viewing. You can see that, that 062 and 015 are still quite distinct from each other, whereas 013 no longer sits up high with the rest of the blues, and the others that we are now considering to be cyanomontanous, even though they're red, because they fit historical morphometric classification. Let's take a look at the admixture idea. So, what is a genetic admixture? Let's take a map and find a location where it is predominant in the region. This is a blowout of between Vernal and up near Manila. Vernal is down in the lower portion of the map, Manila up in the upper left-hand corner of the map. US Highway 191 goes north out of Vernal and across the top of the Uintas, forking and allowing you to go west on Utah 44 just before getting to the Flaming Gorge area. SCA 013 is found at the Carter Creek area, a most fascinating location because there are a number of samples that have been collected at this very population across over 40 years. Some have been labeled variety Goridii, some variety Scariosis, and some variety cyanomontanous, and a few not given a varietal status at all. Going southeast, we can go to the area near Baker Springs. They're only 3.5 miles apart, where SCA 042 is at and 041 are at, and you can see a dramatic difference between the blooms. And you can see that as well in the rest of the plant, but I'm not showing that here. That's a very short distance with a great deal of difference between the two different morphotypes of the area. Between the Baker Springs region and the Carter Creek region, they're just under 19 miles by way of the crow flying. So as a result, we have to stop and think about this, that there's a lot of genetic differences going on there, and it shows up both in the genetics and in the morphotypes of the plants. One of our objectives has been to identify morphometric characteristics to help us separate the differences within the Scariosis complex. We looked at 273 herbarium specimens of Alba fluvis, cyanomontanus, Scariosis, which also in that case included Goridii, and the Tabby Mountain material. We grouped them based upon genetics, so the blues were grouped together, the reds and the yellows were grouped together, and the greens were grouped as the Tabby Mountain. We found 8 characteristics: plant height, distal most colline leaf, proximal most bract width, sepal length, corolla length, anther sac length, total flowers per thierce, number of flowers per verticilliaster. After completing our studies, we have concluded that the Tabby Mountain population should be identified. We have morphometric characteristics associated with it, and it is distinct genetically. 2 of the characteristics found in Jolin's Penstemon that should be recognized as the proximal most bract, or the leaf just below the first whorl of flowers, and the highest leaf just under that bract. Both of those tend to be very wide in many of the samples of Jolin's Penstemon. There are other characteristics as well. There are areas in which there is genetic admixture between Joliniae and variety Scariosis. The conclusions of our study: first, variety Albifluvus should be returned to species status. Second, variety Garridii should be folded into variety Scariosis, and variety Garridii will no longer be recognized. Variety Cyanomontanus has a larger geography than historically viewed. Fourth, there's a large admixture area. We know that the genetics are very blended inside the area of what historically referred to as Garridii. Finally, the Tabby Mountain population is to be recognized as a variety called variety Joliniae or Jolin's Penstemon. Thank you for your time.